Hello students, welcome to biology class. This video is in continuity with the previous video of chapter Transport in Plants. The objectives that we are covering in this video are absorption and uptake of minerals by the roots, translocation of mineral ions, phloem transport, flow from source to sink, and mass flow hypothesis. Starting with the pathway of minerals. Minerals enter the root by two pathways, by active transport or by passively. By active transport into the symplast of epidermal cells, then moving toward and into the stele through the plasmodesmata connecting the cells. By passively moving through the apoplast until they reach the Casparian strip where they are transported into the cells of the endodermis. In both the cases, minerals enter the water in the xylem from the cells of the pericycle through specialized transmembrane channels. There is also exchange of material between the xylem and phloem. Therefore, mineral elements pass up xylem in both inorganic and organic form. They reach the area of their sink, namely young leaves, developing flowers, fruits and seeds, apical and lateral meristems and individual cells for storage. Minerals are uploaded at fine veins ending through diffusion. They are picked up by the cells through active uptake. There is also remobilization of minerals from older senescing parts. Nickel has a prominent role in this activity. The senescing leaves send out many minerals like nitrogen, sulfur, phosphorus and potassium. Elements incorporated in structural components are, however, not remobilized. Example, calcium. The remobilized minerals become available to young growing leaves and other sinks. Though it is generally considered that xylem transports inorganic nutrients while phloem transports organic nutrients. The same is not exactly true. In xylem sap, nitrogen travels as inorganic ions as well as organic form of amino acids and related compounds. Small amount of phosphorus and sulfur are passed in xylem as organic compounds. Next come translocation of organic food in plants. Translocation is the movement of organic food such sucrose and amino acids in phloem from region of production to region of storage or regions of utilization in respiration or growth. Carbohydrates are transported through a plant in the form of sucrose, glucose and proteins as amino acids. Substances can be transported in any direction in phloem. From photosynthesizing leaves down to roots for storage, upwards to growing buds, flowers, leaves, fruits for respiration and growth, and from storage organs such as root tubers to all parts of the plant. Glucose, the product of photosynthesis, is very important as it makes many other important nutrients. Example, sucrose. Sucrose in the leaves then enter the phloem vessels. The phloem transports the sucrose all across the leaf where it can be made used of. From this image we can understand the phloem transport, that is, flow from source to sink. Source refers to the part of the plant that synthesizes the food, that is, the leaf or the green part of the plant, and sink refers to the part of the plant that needs or stores the food, which could be any part of the plant like stem or root. Next is mass flow hypothesis. The mass flow hypothesis of transport in phloem is as follows. Mass flow hypothesis proposed by Munch in 1930 explains the movement of sap via the phloem. When there is a high concentration of sugar present in the source, then a diffusion gradient gets created between the sugar source and the sugar sink where the sugar is stored. This is responsible for drawing water into the cells from the neighboring xylem. When this happens, turgor pressure gets created. A part of the glucose produced by photosynthesis is converted into sucrose. This is then transported actively to the companion cells and then to the sieve tubes via diffusion. The osmotic pressure moves the sucrose through the sieve tubes toward the sink. When the sucrose reaches the sink, it is removed into apoplast. Water moves out of the sieve tubes by osmosis, establishing a pressure gradient. A low turgor pressure is created at the sink to maintain this gradient. Water passes back into the xylem later again. Thus, the mass flow hypothesis mechanism is dependent on the turgor pressure as well as the osmotic pressure gradient between the source and the sink. This is a diagrammatic presentation of mechanism of translocation by mass flow hypothesis. Through this image, we can also understand mass flow hypothesis. As glucose is prepared at the source leaves by photosynthesis, it is converted to sucrose disaccharide. Now, sugar in the form of sucrose move to the companion cells. From companion cell, it moves to the living phloem sieve tubes by the active transport. This results in the hypertonic condition in the phloem at the source. Water from the adjacent xylem moves into the phloem by osmosis. The osmotic pressure builds up in the phloem. Now, phloem sap moves to the area of low pressure, that is, towards the sink. At the sink, the osmotic pressure must be reduced. Hence, again sucrose is moved out of the phloem sap into the cells by active transport. The cells at the sink use this sugar and convert it into starch or cellulose. So, as the sugar are removed from phloem, the osmotic pressure decreases and water moves out of the phloem. This whole sequence leads to mass flow of sugar through the phloem from source to the sink. Thank you so much.